Very good morning to all of you. I am sure all students have already arrived. Today we have with us Mr. Rakshit Chowdhury. Presently he is a product manager working for a very important company in Chennai, that is Pinco Biotech, and he is promoting a product, a very important product for genomics, that is PacBio. About Rakshit Chowdhury, he has been associated with our programs since last 2 years. He was delivering a wonderful lecture last year, and that only prompted me to invite him again to address the students. Rakshit Chowdhury is taking care of genomics and bioinformatics wing of Pinco Biotech, and he is having bachelor's degree in technology in computer science and also additional degree from Institute of Bioinformatics and Applied Biotechnology, IBAB. I guess it is in Bangalore. And he has got 8 years of experience with different products. In particular, the products which is dealing with the genomics, data analysis, and product development also. And prior to his present employment as a product manager in Pinco Biotech, he was associated with 2 other companies in our country. One is Imperial Life Sciences, I think more than 4 years, and he also worked with another company for a very short time, that is Garnet Laboratories, and again in Bangalore. And I was a bit surprised to know that he had a stint of very brief time with the IGIB, that is one of the CSIR institutes in our country, as a senior research fellow. I am sure Rakshit Chowdhury would deliver an excellent lecture on SMRT sequencing, a sequencing which is going to change the face of genomics in the coming years. I call upon Rakshit Chowdhury to deliver his lecture.
Good morning, everyone. Am I audible? Yeah. Thanks, Dr. Kumar, for inviting me again. And this is one of the best structured program for students and research associates I've come across in the country. So I'm more than happy to come here again. And the emphasis here every time has been more towards educating the research professionals rather than what we do generally, that is promote our product. So what I always try and do is start from the very basic. I know you guys have been through this day in day out, but where I want to start is from the very basic, and that is so imagine you have to read a book, and so for that somebody has to write chapters. To write chapters, somebody has to actually form paragraphs, sentences, words, and to begin with alphabets. So sequencing is something which is almost equivalent to having your ABCDs. So what is actually sequencing? It's just reading your DNA in the form of A, T, G, and C, so that we can put it together in a format where it's understandable, whether it is in terms of genomics or proteomics or any other such functionalities. So the whole idea behind doing sequencing is to later on understand the functionality of that particular genome, how is it working. But for that you would need the basic sequence of that particular organism. So the evolution of sequencing was so it started somewhere roughly in 1977, where people were thinking about the human genome project, and all the Sanger thing came into picture. So this is the evolutionary pipeline of the whole sequencing thing. So major were 1977, then somewhere in 2003 human genome project was completed, double quotes, because I still believe it is not yet fully finished, but because of the chemistry available at that particular time, this was the best possible genome we could get. Later on, Roche pyrosequencer came in, Illumina's sequencer came in, then came single molecule real-time sequencing, which is the long read sequencing. Then we have now a few more of these long read sequences out in the market. So why we need all these different kind of sequencers? So what happened was, Sanger is still gold standard if you want to revalidate something, but it's terribly slow and it's very, very costly if you have to do a big genome. So what people thought, why not have something which produces tons and GBs of data in terms of sequencing, in terms of ATG and Cs. So that's why the idea of short read sequencer came up, which was also known as the second generation of sequencing. When that thing came up, people got a lot of data. People got a lot excited. But at the end of the day, they started realizing that what we are doing is just accumulating a heap load of ATG and Cs, and we are not able to assemble them correctly. It was like you already have a magic book, but you cannot make sense out of it. So ultimately, something has to be done to move one step ahead and understand the magic words written in that book. Then came the single molecule real-time technology, and why we call it single molecule real-time, because one single molecule is being added in the real time every time a base is being read. So it's a in vivo process happening in vitro. Same way it happens inside our body, exactly same way it happens inside a sequencer. We have just slowed down the polymerase. So this is the kind of polymerase we use. What happens is the template DNA goes into your polymerase. One base is being added. There is a camera at the bottom of this particular event which captures this particular event, and one base is, so approximately 2 to 5 bases are added per second, and all this is captured by a camera in the form of a movie, which in turns get converted in the form of pulses. So all these pulses have a different demarcation, not just in color but also in terms of time, and that is how we convert it inside a sequencer into A, T, G and C. So what was unique about this particular sequencer which was not there in the other sequencers? Here the read lengths were really wrong, and when I say really long, the average were more than 30,000 bases, and we now go as high as 500,000 bases. That's like half MB. We had a very high accuracy level, so we were not just going vertically, we were not reading just vertically too many bases, we were very accurate at that particular time. We have now increased this to way above. So this is like one error in 100,000 bases, that is 1 lakh bases. So usually in our body there is an error in 1000 bases which our polymerase has, but that is corrected. This also is corrected at some fold coverages. Like other sequencing technologies, second generation faced a big issue that whenever you used to do amplification, there was a lot of GC bias, and the way chemistry used to work, again, whenever there used to be a stretch of GC, we were not able to read that particular sequence correctly. So in this, which I'll further discuss in detail, in this we were able to overcome that particular challenge of reading those GC or AT rich regions. Also, without any additional protocol, we were able to decipher the epigenomic information, 6MA, 4MC, 5MC, and other such DNA modification events without having an additional protocol. So in second generation, what used to happen, we used to have a bisulfide sequencing protocol, which is very tricky, and here we just do the sequencing and we get this additional information. So that was, we never aimed for this, but then we realized we are getting this information because what we have is file in terms of pulses, and we also know how much time one pulse takes. So whenever a pulse used to take a little longer, we recognize those pulses, we got to know what kind of modification is happening, and that is the reason that's why the pulse ratio is now different. So majorly these three things were the things which Specific Biosciences Smart Sequencer SQL is now covering. So these are two major applications, whole genome and targeted sequencing. Under whole genome, we have genome assembly, we have SNPs, structural variations, transcriptome, base modification. Under targeted, we have long amplicon, we have HLA, we have target capture, we have 16S, minor variant, and also we can do multiplexing. Anyone can let me know what multiplexing is? Any idea? So multiplexing is, what we do is, instead of just running one sample at a time, what we do is we run more than one samples in one experiment. So we have barcodes. So you must have seen barcodes everywhere on your, on your garments, on your biscuit packets. So just like that, we have barcodes made out of ATGC. Those are unique. We attach them with a particular sample, we sequence it, and when we have the sequence output, we decipher it, and we call that process as demultiplexing. It means we chop out those particular, we bin those particular samples together which belong to one particular organism. So the major use of multiplexing is that it saves time, it saves money, and you can actually do multiple kind of samples together. So again, because I wanted to touch upon basics, there was a lot of confusion when 1000 Genome project came up, a lot of confusion when a lot of papers were coming out of genome assembly. The confusion was people used to say, yes, we have a complete genome, but it's a draft genome and it's 87% complete, 80% complete, 78% complete. So people were confused. If it is either complete, it's a 0 or 1, right? It should either be completed or it should not be completed. So what is this? So what used to happen was, because the technology at that particular time, in that particular era, was not that advanced that it used to finish off the genomes completely, so the genomes were not closed. So what used to happen is, there were a lot of ambiguous bases. So when you talk about contig, a contig is continuous bases. So you will not find any ambiguity in these sequences. You will only have A, T, G, or C, all the known 4 bases. But when you talk about scaffold, you will have a 5th base called N, and N means an ambiguous base. It can be anything. It can be A, T, G, or C. So this was a shortcoming of not just the chemistry but also the algorithms, that it was not able to decipher what this thing is, and this was majorly because of the factors like GC bias, the nucleotide composition of the genome, because there were genomes. So to be honest, no offense to the human guys, but human genome assembly is much more trivial as compared to plant. You have a lot of ploidy, you have a lot of heterozygosity. Plant genomes are, even if the genome size is small, it's way more complex than any anywhere when you compare it with a human genome. So we used to see a lot of such genomes coming out, especially in plant, where a lot of ambiguous bases were there. So if you have just 70 or 80% of the information from a genome, that means you do not have your 100% information. And when you do not have 100% information, you might be missing onto those important functional genomics aspect which you are keen to do. So for example, what you ultimately want to do is, say suppose there is a breeding project where you want to find out those single nucleotide polymorphism or point mutations which are responsible for a particular trait in a plant. But what is happening is, because that particular trait is lying in a genomic region which has a lot of complexity, and when I say complexity, it means it can have a lot of ATs together, it can have a lot of GCs together, or it can have a lot of homopolymers, AAA, TTTTT. So because of the sequencing technology, we were not able to decipher those. And when I say it was not able to decipher those, it was because these regions were in kilobases. And the reading technology which we had, they only used to produce reads of 100 bases, 150 bases, 200 bases. So ultimately, all the burden will be all the algorithm to stitch them together in a line. At the end of the day, algorithm will not know these are coming as in one after the other, or these are